Because, of course, what we really want to do is we want to be able to surgically go in, find someone's genome in which there may be hundreds of different typos in that genome, only one of which causes a disease, and we want to be able to go in and surgically replace just that one mutation. Maybe even diagnose people that have different genetic diseases. If we could eventually scale up to some sort of speed in which we can say, here are a whole bunch of variants of unknown significance, and maybe in a cell culture model, we can put all of those into those cells and figure out which one is causative of phenotype, that can be useful. So I think that we both have the desire to go backwards, right, to be able to take causative mutations that we know are causative and cure those, as well as to be able to model things very rapidly. But of course, everything comes down to DNA repair, right? All we do as editors is induce double-strand breaks in genomes, and everything else that happens is up to the cell that it's in. And I think that really puts light to some of the things that have been, at least in my experience, a little bit frustrating about genome editing. For example, one of the things that works very well for genome editing right now is knocking out genes. You can induce a double-strand break, and at least in human cells, non-homologous end joining and alternative end joining pathways are very powerful. So you can, in certain models, certain targets, certain loci with CRISPR, you can get literally 100% of alleles knocked out through sort of error-prone repair of the site. When it comes time to put in edits, this process is much less efficient. And one of the things that I'm going to end with is sort of telling you new ways that we've gotten insight into how this process is going on, and maybe with some implications for therapy. And hopefully, it'll eventually lead to some ways that we can improve the process. So, of course, knockout is all well and good. It lets you figure out, what does this gene do? But very often for therapies, what we want to do is we want to edit genes. We want to take a donor DNA, and we want to have some sort of information in there, and we want that precisely inserted or to modify a site. I want to draw a distinction right now between two different types of donors you might use. There are other types of donors, but for now I'm just going to talk about the difference between double-stranded DNA donors like you might have on a plasmid or something like that, and single-strand donors, which for the rest of this, and for my purposes, are going to be oligos. So you can, there's a big difference in how these guys are constructed. Double-strand donors tend to be very large. They have many kilobases of homology. Single-strand donors are something that we've been using a lot in my lab because they're quite easy to get. They're quite easy to use. They're literally just ultramers from IDT. You can protect them. You cannot. And so that gives you a lot of scale and a lot of speed. So instead of needing to make large, complex vectors every time you want to do an HDR experiment, you can literally do hundreds of different types of HDR experiments. And I think that really brings what I would call editing, where you're putting in this type of information into the same type of scale, the same type of speed that we now have the ability to make for the cut with CRISPR reagents. I just want to point out that this whole theme of everything downstream being a product of the cell is not restricted to cutting because, of course, CRISPR can be used for transcriptional regulation. If you mutate out the active sites, you can recruit transcriptional repressors, transcriptional activators, but these are typically cell encoded proteins. So you have, say, an effector domain that recruits repressors that are present inside the cell. And so you can turn down and turn up gene transcription, but you're relying on everything that's in that cell to actually make that happen, okay? So these are really transformative homing tools, Cas9 is, and other types of Cas enzymes, but we're really still relying on the basic biology of the cell, okay? And what that means is that editing and regulating genomes is really action at a distance. So instead of being this kid who's got a pen and is holding it, it's really a little bit more like this machine from the 1800s called the polygraph. Thomas Jefferson owned one of these. He used it to copy documents. And really what you're doing is you're writing on one side and hoping that the cell makes the change that you want on the other side, right? We have things that we want to enact. And we really have to rely on some sort of complicated mechanism that connects these two things, what we're trying to have happen and what actually happens in a cell. Okay? So I think that explains a little bit why things are challenging at first, because especially if we're talking about genome editing, to say that we have complete control and complete understanding of everything that's going to happen after an edit is to say that we have complete control and complete understanding of DNA repair, everything in between. And of course, that's not true. There's still a lot to learn there. So that's one of the things that I'm going to talk about today. I'm going to start, though, with just sort of an applied example of how we can use genome editing. I'm not going to belabor the biology of this because I'm going to assume for everybody in this audience this is very old hat. This is something that it's a disease that we got into through work with collaborators, Mark Walters and now Don Cohn. We really got into gene editing for sickle cell disease. So sickle cell disease is where a single point mutation causes hemoglobin to polymerize, causes red blood cells to adopt a characteristic sickle shape. They're fragile, they're stiff, they clog arteries. It has many terrible complications ranging from pain all the way to anemia and organ damage. Even in the developed world, life expectancy is greatly decreased. In the developing world, it's far, far lower. In the US, there are about 100,000 people with this disease, and of course, millions more in sub-Saharan Africa. So I think that what I'm going to be telling you about is trying to use genome editing to surgically correct the causative mutation of this disease. But I think that, as you'll see, what we're talking about here is so-called ex vivo editing, where we take cells out, we edit them, we put them back in. And I think that there's a lot of opportunity for that in the developed world. But if we start to think about treating millions of people in Africa, I think that there are plenty of people in the field that have some really amazing ideas about ways that this can be scaled. And I think that ex vivo editing is probably not the way to go there. But the thing I want to point out is that care for this is supportive. Early mortality is really the norm, which is especially frustrating because the point mutation, the molecular nature of the disease has been known for about 70 years. If you have a homozygous mutant, you have the disease. If you're a heterozygote or wild type, of course, you don't have a disease. So from an editing point of view, all we need to do is just convert one allele. If we get two, that's great. But if we convert just one allele, that should cure people of the disease. You can use allogeneic bone marrow transplant for this. It does cure people. It's relatively rarely used. It can be kind of hard to find donors for this. Ablating the bone marrow leads to fertility problems. People tend to avoid the allogeneic bone marrow transplants for that reason. There's also a small risk of rejecting the graft. So people sort of weigh the risk-reward in which this is not a disease like severe combined immunodeficiency in which you need to get a bone marrow transplant absolutely right now. It's more that your life expectancy is severely shortened, and so people weigh that calculus, and typically bone marrow transplants are not performed. So of course what we want to do is we want to change that calculus, and we want to make it so that we can transplant a patient's own edited tissue to make it much more favorable. So what we do is, of course, probably nothing new to people in this audience. We plan to take tissue out. We would want to edit it, in our case, using CRISPR-Cas9 types of reagents to cure the disease, put it back in. We can get some idea of how much editing is necessary to do this, and I think it's really important to point out that that number is not 100%. So there are people who have received allogeneic grafts who have been cured of the disease by having far less than 100% graft. So from mixed chimerism, it suggests that with as little as 10% or less of donor tissue, healthy donor tissue, you can cure the disease. And this in part comes down to the circulation lifespan of the red blood cells. Sickle red blood cells last about 20 days in circulation, wild type about 120 days. So I think it's important to point out we're not aiming for 100%. If we can get as high as possible, that's great. But we don't need to get 100%. Now the problem is we need to be making a change that affects red blood cells, which of course have no genome, they're anucleate, and they have a relatively short lifespan in circulation. So to make a long-term change, we need to be editing all the way back up the differentiation chain to edit long-term repopulating stem cells. The problem is these, of course, are unculturable. Every time we work with them, we get them from donors, primary tissue, and in the past they've been somewhat difficult to edit. When we eventually get going in this, there's a variety of criteria we have for doing this. Personally, in our team, we want to make as small a genetic change as possible. We don't want to have any kind of selectable marker in there. This is mainly trying to keep the edits as innocuous as possible because, of course, we know exactly what the outcome of reversing just the causative SNP is, but putting other markers in, it sort of adds a little bit more uncertainty, so we try to avoid that as much as possible. We want the highest possible efficiency. Mixed chimerism suggests that we need to be 2% at the very, very, very lowest, 5% we'd be happy with, 10% or more we'd be absolutely thrilled. We want the fewest possible off targets, but I think one thing that I would point out, there are a lot of ways right now to off targets. And those are luxuries that no other therapy really has, right? You don't take a large molecule, say an antibody, and say, in the entire proteome, what is every single protein that it binds? You instead ask, what's the functional tox of that molecule? And I think that's something that will, at least the messages we've gotten from regulatory agencies, is that functional tox trumps long lists of off targets. So I think that we want to be aware of what those off targets are, and I think we need to measure them and know what they are. But I think that just having them without having some sort of measure of toxicity is not sufficient. And then finally, just a couple of small things. We need some sort of edit that's not toxic to the long-term HSCs themselves. We need the edits to appear rapidly because we have a limited amount of time to work with the cells. And we want the Cas9 and editing reagents to be cleared pretty quickly. That is to say, we don't want to put the reagents into the cells, have them stick around for a long time, put the cells back in, and have the editing reagents still in the cells, still possibly hitting something that we haven't measured yet. So we got started with this with a really fantastic postdoc in my lab, Mark DeWitt. This is a great collaboration with Dana Carroll, Dave Martin, and Mark Walters. So we're going to take the locus, we saturate every single guide RNA for Cas9 that we can find in the site, and we use single-strand DNA donors to edit this. So we go through guide RNA screening in cell lines. It's pretty well established now that certain guide RNAs work better than others. A lot of debate around why that is. People have found sort of machine learning ways to figure out what's a good guide, what's a bad guide, but a lot of times those machine learning approaches don't tell you why it's a good or a bad guide. So we tend to just screen through every guide, try to find the ones that work. We then optimize, work for that in HSPCs, and then we go into mouse models. So we've decided to go for using Cas9 RNPs. So this is the so-called ribonuclear protein complex. So here we purify recombinant Cas9 protein. We use T7 transcribed or synthetic guide RNAs. We form a complex, and we pair that with single-strand DNA donors, and we get them into cells by electroporation. Part of the reason we use this is that it seems to be very effective in a whole variety of contexts. I'm going to be talking about HSPCs, but it also works in a whole variety of different primary cells. And the efficacy is extremely high. The other really nice thing about this is the scalability. Just to let people get started, there is protein available from something called the Berkeley Macro Lab. If you don't want to purify your own Cas9, this is available for actually quite cheap. I think it's $2 per editing reaction, something like that. There are protocols here that my group up for making these transcribed guide RNAs. Those are at scale. A single graduate student can make 500 of them in 2 days. So it's really, really fast. And it's actually very, very inexpensive to do. So the scalability means that, again, we can just saturate every single guide RNA and explore every single donor in this region. This is generally what this looks like. This is why we screen different guide RNAs. This is a T71 digest, just looking at with and without T71. Skipping over how T71 works, you're just looking for the appearance of bands underneath this amplicon from genomic DNA. You can see that different guides give you different efficacies. So these dark bands mean that the guide worked really well, and these guides didn't really work well at all. So we quickly homed in on something that we call guide 10. We quantified, at least in cell lines, again, how well this works to program edits, not just making error-prone mutations, but actually programming the sickle change. And we can see that we can get very high efficiencies, up to about 40% allele conversion, HDR in cell lines. So we move to HSPCs. We take so these are our HSPCs from healthy individuals. So at the moment, right now, we're putting in the sickle mutation. So instead of taking out the sickle mutation, we're putting it in because we can get access to the healthy tissue much more easily. Okay? So it's a single base difference between these two different donors. We quantify by sequencing how much allele conversion we get. We can get, again, up to around 40% allele conversion of the SNP by sequencing. So we take those edited HSPCs and we put them into immune-deficient mice. We let them go for 4 months, and we saw something really interesting. So on the one hand, we saw actually a good amount of HDR. So the best we were able to achieve, this mouse has around 6% HDR, lasting for about 4 months. We're right around or a little bit above where we might get benefit from mixed chimerism. We'd like to be higher, and that's what we've been optimizing lately. Right now, we're instead more around the like 10 or 12% mark. In mice in this experiment, we're more around like the mean 3%, max 6%. But one thing that we saw that was very interesting was that in these mice, the indels that come along for the ride with the HDR persist, but somehow we lose the HDR over time. And that was really strange because that suggested it's not like we were killing the HSPCs, it's not like the electroporation was doing anything bad to them. Whatever we were doing was not overtly toxic. There seemed to be something about going through HDR that seemed to cause a loss in repopulation. And so we got really interested in why that was. So that's what I'm going to talk about for the second part of the talk. This has so far been sort of a theme in HSPC editing. So it's now been observed by Paula Cannon, Don Cohn, Matt Porteous, using especially single-strand DNA and AAV donors who tend to lose HDR. Interestingly enough, so in the previous slide, we're just counting alleles, but if you go through Hardy-Weinberg equilibrium, that would tell you that more cells will have at least one allele than there are numbers of alleles. And so when we took cells back out of the mice, we plated them on methyl cult, and then counted colonies, sequenced colonies to figure out the zygosity. And indeed, when we're counting numbers of cells that contain HDR alleles, we do better than that 2 to 6%. So I just want to point out that what I was reporting previously is just allele counting, not how many cells contain an HDR allele. And so by the measure of how many cells contain an HDR allele, we're not doing too bad again. Like I said previously, we've now been able to improve this a little bit. So the big question, of course, is function. So here we take sickle HSPCs. Previously, I was showing you taking wild type, putting a sickle mutation in, because those are the easiest cells to get. We then managed to get sickle HSPCs. This is from Mark Walters, Children's Hospital Oakland. We edit them, we differentiate them into red blood cells, and then we use RNA-seq and HPLC to quantify globin abundance. And as you can see, this is someone with sickle cell disease making all sickle globin, no wild type, and then after editing, they now make more wild type globin than sickle globin. So we think this is really promising. We are able to achieve some amount of editing in human HSPCs. Those edited HSPCs do persist over a long period of time. We do get this switch in globins. We've been now working on IND-enabling studies, again with Mark Walters, Don Cohn, through great generosity of the California Institute for Regenerative Medicine. But we're left with this really fundamental question of why is it that the indels stick around but the HDR is lost? And as we were thinking about this, we came to the realization that people have talked a lot about this bifurcation between indels, NHJ, and HDR, but it really wasn't clear what was happening in that black box in HDR. And especially because we're using single-strand templates, it could be that these operate in a very unusual way. So digging around, part of my background is in DNA repair, it suddenly became clear that when people talk about doing editing using double-strand templates, they're most often talking about homologous recombination, which is the type of HDR everybody normally thinks about. It's Rad51 mediated, you've got strand invasions, holiday junctions. Think back to what long ago how you thought gene replacement was going to work and the way stuff works in meiosis. But what's happening with single-stranded DNAs? How is it that single-strand DNAs give you editing? How could that be Rad51 mediated? Because when you're using duplex DNAs, you have these long stretches of homology. You require about a kilobase. SSODNs can be as short as 100 bases, right? There's no holiday junction that might be formed. There's no duplex. So how is it that these work? So this was a really fantastic work by postdoc Chris Richardson. And really what we just asked was, what's the difference between double-strand repair, which is relatively inefficient, even in cell lines, giving you maybe 1, at most 5% HR, and is active in G2M, and this thing that we're calling SSTR now, single-strand template repair, using these SSODNs. So this can be very, very highly efficient. I showed you 40% in HSPCs. The best we've gotten in cell lines is around 60% of alleles converted. And this seems to be quite different mechanistically. Number one, that even if you're editing the same locus, as I'll show you, you get very different efficiencies. And number two, they're active in completely different phases of the cell cycle. This is sort of literature data from the labs of Jennifer Doudna, Maria Jason. These seem to be operating in very different ways. So we first thought, well, can we just look and just make some educated guesses about what might be doing this? And we quickly got overwhelmed because DNA repair is sort of this dizzying process involving hundreds of different factors which kind of overlap in pathways. Some of these factors get reused in some pathways and not in others. And so we quickly sort of got a headache thinking about this. So we started to ask just the first question, is single-strand DNA repair HR? And what I'm going to convince you of now is that it is not. Okay? So like I said, classic homologous recombination relies on Rad51. It's completely dependent on it. Okay? If you do a single-strand DNA editing experiment with a reporter in which you're turning BFP into GFP by a 2-amino acid mutation, it is completely independent of knockdown of Rad51. So I'm not showing the qPCR here, but we've got upwards of 90% knockdown of Rad51. By contrast, if you SI and do the exact same editing experiment with a double-strand DNA plasmid, it is dependent on Rad51. Okay? So single-strand DNA donor editing and double-strand DNA donor editing seem to rely on different sets of factors. Okay? So this core thing, which should be involved in both processes if they're both HR, is only required for double-strand plasmid editing. Okay? So to try to figure out, rather than try to go through one by one through that dizzying array of factors, we decided to take a more unbiased approach. So what we did was we took a library of guide RNAs that are programmed not to knock out alleles, but to knock down alleles. So here we're using so-called CRISPR-I. This is a technology to decrease transcription of a gene. Okay? So we can multiplex this hugely. We take thousands of different genes in this guide RNA library. We knock them down such that in each cell, only one gene is knocked down. Okay? Then in the context of that knockdown library, we make a single edit at a reporter locus. Okay? So we make an edit, and that reporter, we can monitor a couple of different outcomes. We can look to see whether this locus is fidelitously repaired or unedited. We can see indels, error-prone repair, and we can see SSTR because we convert to GFP. And because we have this color change, we can collect different bins, and we can do a FACS experiment. We can compare relative abundance of these guide RNAs to figure out which genes are required for which type of repair, and even which genes give you upregulation of different types of repair. I'm going to focus on which genes are required for different types of repair. So this is a volcano plot showing log 2-fold change in guide RNA abundance for different genes versus the p-value. And the first thing that popped out to us, on the left, these are genes that are depleted for single-strand template repair. That is, they are required to do the process. Black is negative control guides. These are non-targeting guides. So that sort of gives you an idea of the background. And up above the background, everything in blue here, we were shocked to see, because this is basically the entirety of the Fanconi anemia pathway. Okay? So what this first told us, surprisingly, is that single-strand template repair seems to not be homologous recombination, but it is instead FA. So what is FA? Fanconi anemia factors are so-called because of the genetic disease that their loss causes. It's a very large multi-protein and multiplayer process that's involved in recognizing interstrand crosslinks or stalled replication forks. And it has a variety of complexes that are involved in recognizing the problem, assembling a core complex to do something about it, removing the crosslink, and then actually going through and doing the repair. Okay? And what we found is that members of every single part of this process are lost from the guide RNA library when we try to do SSTR. Okay? So Fanconi anemia genes acting in every part of the pathway are required for SSTR. We went ahead and we did individual knockdown of each one of these, as well as cDNA complementation. Every single one of them is required. So you require members of the core complex, things that are involved in recognizing the cross-link, things that are involved in recycling the cross-link repair factors. Every single one of them is required, and in the context of knockdown, re-expression rescues the phenotype. This works across multiple loci, works in multiple cell lines, and also works in primary cells. But strikingly, the Fanconi anemia pathway does nothing to indels at all. So I'm showing you sequencing results from non-targeting versus knockdown of FANC-E or FANC-A. This is just looking at indel distributions. You can see that absolutely nothing is changing to either the frequency or the molecular outcome of those edits. We have a couple of SNPs when we do this SSTR to be able to follow at different distances from the cunt site. And the thing that I want you to see is that knocking down Fanconi anemia factors knocks down the frequency of all these SNPs equally. Okay? So we're not so-called changing conversion tract length. We're just taking the entire process and muting it by knocking down these factors. So what this really tells us is that what we've been thinking we were working with all along was homologous recombination. This is why people have typically used HR activating factors to try to increase HDR. People have tried to use RAD51 activators, people have tried to use 53BP1, people have tried to mess with the cell cycle, and it's had limited effect. So it's been useful in some cases, but it hasn't been totally a game changer. And one thing that I might leave you with today is it could be because we've been looking at the wrong pathway entirely. Okay? At least for single-strand DNAs, it seems like instead we need to be focusing our attention on the Fanconi anemia pathway. It's a little unclear right now molecularly how the Fanconi anemia pathway is enacting this type of repair, but it's very clearly the responsible player. Final thing I'm going to leave you with is that this has really big implications for therapeutic gene editing, especially for the HSC stuff that I was telling you about before. This is a mouse model of Fanconi anemia. If you induce DNA damage, the hallmark of Fanconi anemia is loss of hematopoietic stem cells, loss of long-term hematopoietic stem cells. Okay? That is the fundamental, that's why they're called Fanconi anemia factors. It's the fundamental clinical phenotype. And so we're left with this question, could it be that the act of going through Cas9 SSTR is the very thing that's causing us to lose HDR alleles? Okay? Could that be why we're losing the HSCs? Could we maybe re-express Fanconi anemia factors or find some way to transiently get them into HSCs to drive higher repair in HSCs? Can we maybe protect HSCs from these types of effects? And then anecdotally, we've seen among different donors, among different cell donors, we've seen very marked differences in SSTR frequency. And so we're now starting to wonder, could it be that the expression levels of Fanconi anemia factors is a biomarker for editability? That is, people with high expression levels of all of these factors will be easier to edit, and people that, say, have lower expression of one factor or another may actually be more difficult to edit. And then on the very extreme of that case, these results would suggest that you would not be able to use single-strand DNAs to cure Fanconi anemia by gene editing. You would have to use a different donor type to do that. Okay? So those are the thoughts I want to leave you with. I just want to finish up by thanking the real heroes of this work. Again, it was Mark DeWitt who did all the Sickle editing, Chris Richardson who did all of the Fanconi anemia work. Thanks very much to really wonderful collaborators, including the labs of Dana Carroll, David Martin, Don Cohn, Mark Walters, Jonathan Weissman. Thanks very much to funders. Thanks to you for your attention, and I'll take any questions.
So for the questions, we'd like to ask everybody, please step up to the microphone since we live stream this session to the outside. So please step up to the microphone. And also we'd like to ask the people standing in the back, either move to the front, and everybody who's sitting on the outside, please move to the inside. There's lots of people at registration, and we'd like to have everybody seated. Thank you. So maybe our first question, I noticed there were also proteins of the SLX4, SLX1 complex. Have you looked at the factors of this pathway, like EMIA1 or MUSID1? Because they're formed this very kind of multifunctional kind of nucleus complex, which would be really perfect to process sort of an integrated single-stranded oligo at the site. Yeah,
that's a great point. So yes, MUSID1 is required for this process. So what we think is happening is not that Cas9 is itself a roadblock to act as a crosslink, because like you saw, NHEJ does not require Fanconi anemia factors, right? So what we think is happening is instead that having the single-strand DNA there acts sort of like a stalled replication fork, which Fanconi anemia factors are also required to repair, and that some sort of intermediate involved in single-strand DNA invasion is triggering recruitment of Fanconi anemia factors, recruitment of MUS81, SLX4 repair complexes.
Thank you for the exciting talk. Often single-stranded RNA is used for the CRISPR-Cas technology. Do you think it's processed the same way?
So I think what you're referring to is people use single-strand RNA that is mRNA to introduce the Cas9. Is that what you're referring to?
No, they just, they do transfection with, say, mRNA with Cas9 and the single-stranded RNA.
For the guide RNA?
Yeah.
Yeah. So you have to have the guide RNA to program the edit. I think if you just have Cas9 that is with no donor, you don't require the Fanconi anemia factors, right? This is only when you're doing templated repair. Now, I think what you bring up raises an interesting point. Francesco Storici has some really interesting data showing that mRNAs can template repair in yeast. It's relatively inefficient, but as far as I know, no one at least has reported in a genome editing experiment using an RNA to template repair. But it's a really interesting idea to try that. The prediction is it should work given Francesca's data.
Thank you.
So if I understood your data from mice correctly, it was your HDR alleles weren't persisting over time relative to indels. Is that correct?
That's right. After about 4 months, they seem to stabilize. So they come down to around 2 to 6%, and they kind of stabilize there. But we start off, immediately after editing, we have equal amounts of indels and HDR. And then over the 4 months, those diverge. So the indels stay at 40%, and we come down to sort of a stable 2 to 6%.
But if you're saying that the FA, like overexpressing FA alleles would help with a higher incidence of HDR, what would be the mechanism for FA helping the HDR alleles to persist?
So this is very much a speculation. So the idea would be that if you have some sort of cell context in which now these are two different problems, right? One being some cells we've seen, I didn't put the data up, but we've found that some cells, some cell types, and even just cell lines between the same cell type are not editable with single-strand DNAs. That is, you'll try to edit the exact same locus, and in one cell type you'll get great editing, and in the next cell type you'll get absolutely nothing, even in a cell line, right? And so one possibility is that it's differential expression of Fanconi anemia factors. From some RNA-seq data that we have, it seems like that could be the case, but at this point we're comparing relatively few numbers of lines, so it's hard for us to make some sort of statistical argument that this is the thing that causes it. So we're at the moment going back into some of these cells that don't do single-strand DNA repair on their own. We haven't knocked anything down, and trying to do this complementation to see if that's sufficient to re-energize them for HR or HDR.
One quick follow-up: do you have any comparative ability to knock in single-stranded DNA with a SNP versus a larger cassette?
Larger cassette, yeah. So this is not my data, but there have been a couple of really interesting bits of work by people who are putting larger cassettes in. So I know Sylvie Gouchard and Guru Murthy have different ways of putting large cassettes in. And IDT has recently started selling 2 kilobase long single-stranded DNAs. I think those show a lot of promise, especially if you can put in those big cassettes, you may even have the ability to select something out of that. Putting in selectable markers is not something that we particularly are interested in for our biologies, but I think that that does offer a lot of promise, because then you don't have to be spending a lot of time making these large double-strand DNA plasmid donors where you're PCRing out 2 kilobases of genomic DNA to put in a vector. You just go to IDT, order your donor, and get it in a week. So I think that some of those things have people are interested in them for good reason.
Great talk and great screen. I was wondering if you try to go after also the genes that are involved in HR with the plasmid system. This is the first one, and the second one is, so the screen was done in cell lines, whether you confirmed also these genes directly in the primary hematopoietic stem cells.
So two questions. The first one is, have we done this screen in HR? The answer is we're in the process of reading that out right now. The problem is HR is a much less efficient process. I didn't point that out too closely, but HR, typically we get like 1 to 5% of alleles go through HR. SSTR, it's more like 30%. And so the number of cells we have to run through the sorter to do the HR experiment is, 15 times more to get the cell coverage. So that screen is done. We're reading out the data right now, and maybe next ASGCT I'll tell you the results of that. And then the question of whether or not we can do this in HSCs that have FA, we tried. The problem is we can't get FA HSCs because, of course, FA is a gene of stem cell repopulation. So we tried really hard to find people that could. Because what we want to see is some difference in SSTR between, of course, wild-type HSCs and FA HSCs. But the problem is we can't get FA HSCs because FA is itself a disease of long-term stem cell repopulation. So it was sort of a chicken and egg thing there, which is why we worked with instead these neonatal fibroblasts in which we can knock down the FA factors. But if anyone in the audience that has ideas or sources for how to get FA HSCs, I would love to talk to you.
I'm afraid we have to move on.
Okay.
Not sure this microphone works. I will try here. Okay, it's my pleasure to introduce the next speaker, Daniel Bauer. He got his MD PhD degree from the University of Pennsylvania. He's now at Boston Children's, and he will talk also about editing to cure hemoglobinopathies. Thank you very much for jumping in here.
Great. All right, good morning. So I'm tasked with you to tell you about how we can move editing towards the clinic, and I'm going to focus also on the beta hemoglobin disorders. Here are my disclosures. Oops. All right, so the beta hemoglobin disorders, sickle cell disease and beta thalassemia, comprise the most common monogenic diseases in the world. And this cartogram illustrates the anticipated global incidence of sickle cell disease, and the area of each country is proportional to the number of sickle births anticipated in coming years. And you can see the immense nature of this problem, particularly in Africa. And while the beta hemoglobin disorders are really like the prototypes of molecular diseases where we have deep understanding of the genetic and biochemical bases of these diseases, these are quantitative or qualitative disorders of the beta globin gene that are therapeutic options remain quite lacking, that we have transfusions and chelation, we have antibiotics, we have hydroxyurea. When we think about curative approaches, the current therapies rely on the transplantation of allogeneic stem cells, but this has challenges. There's a limited pool of donors available, there's immune incompatibility, and transplant-related morbidity and mortality. So an alternative strategy might be to collect autologous hematopoietic stem cells from patients themselves, modify those cells, and then reinfuse them. So those modifications could be varied. They might consist of conventional gene therapy with addition of the globin gene. They could rely on correction of the underlying mutation, such as the work that Jacob's just presented. Alternatively, there's a range of approaches that rely on the induction of fetal hemoglobin. So these fetal hemoglobin approaches stem from the clinical observation dating back to Janet Watson in the 1940s that the red blood cells of newborns are protected from sickling due to the presence of high levels of fetal hemoglobin. Then only with time after birth, as fetal hemoglobin levels wane, do the disorders of adult beta globin, sickle cell disease and beta thalassemia begin to clinically manifest. Here, the therapeutic goal would be to turn back on high-level expression of gamma globin, either to replace the absent globin in beta thalassemia or to have an anti-sickling effect in sickle cell disease. So it turns out that fetal hemoglobin levels vary widely among individuals. Here's an old photograph of various red blood cells from individuals stained for fetal hemoglobin, that the levels tend to be less than 1% in healthy people and up to 10% in individuals with sickle cell disease, and that these levels of fetal hemoglobin are largely genetically determined, and that genome-wide association studies are large case control studies that can identify common variants associated with phenotypes such as fetal hemoglobin level. And in this case, the GWAS was really tremendously informative in that just 3 loci explained about half the heritable variation. These included variants at the beta globin cluster itself, at a cluster on chromosome 6 that had previously been implicated by familial studies, where the MIB gene is the actor. And then highlighted here in yellow are SNPs on chromosome 2, marking a gene BCL11A that prior to the GWAS was not known to be involved in fetal hemoglobin control. These genetic studies have been highly reproducible among healthy individuals and in individuals with beta hemoglobin disorders, in whom these same SNPs are not only predictive of fetal hemoglobin level, but also of the clinical severity of the disease. Work from a number of labs, including that of my postdoctoral mentor, Stuart Orkin, have unambiguously shown that BCL11A is a key repressor of fetal hemoglobin. In this evocative mouse experiment, a mouse engineered to have sickle cell disease was crossed to a mouse conditionally deficient for BCL11A in the erythroid cells. And you can see, in the absence of BCL11A, a disappearance of the sickle cells from the peripheral smear, an essentially pan-cellular induction of fetal hemoglobin, and a reversal of the hematologic and pathologic manifestations of disease. This body of work has nominated BCL11A as a rational therapeutic target for the hemoglobin disorders. However, BCL11A plays wide roles in a number of cell types, including important roles in neurons, in B lymphocytes, and in hematopoietic stem cells. We set out to explore the mechanism whereby the common genetic variants at BCL11A are associated with elevated fetal hemoglobin and milder sickle cell disease without any apparent deleterious consequences. So here is shown a schema of the BCL11A gene. Note that more than 50 kb from the transcription start site in intron 2 are a cluster of SNPs associated with fetal hemoglobin level. We compared this to the pattern of erythroid DNA sensitivity, which is an indicator of open chromatin with regulatory potential. These studies were done in collaboration with John Stam at the University of Washington. And you can see that there are discrete peaks of erythroid DNase 1 hypersensitivity at intron 2 overlying the trait-associated variants. Zooming in, note there are 3 erythroid DNase 1 hypersensitive sites indicated here by these colored rectangles as plus 55, plus 58, and plus 62, named based on their distance in kilobases from the transcription start site, and each of which has overlying trait-associated variants. Moreover, in other cell types, like the brain and B lymphocytes that express high levels of BCL11A, there's a paucity of DNase 1 sensitivity in these regions, suggesting erythroid-specific regulatory potential. Along with Crew Smith and Stu Orkin, we characterized mice that had deletion of this orthologous intronic BCL11A enhancer sequence and crossed them to mice carrying a human beta globin cluster transgene. So normally these mice silence gamma globin around mid-gestation, but note in a dose-dependent manner, that the B7A erythroid enhancer is required for gamma globin repression, and that the homozygous enhancer knockouts fail to repress gamma globin. So this result of the enhancer knockout mice phenocopies that of the B7A gene knockout mice with regard to the globin gene regulation. However, unlike the conventional gene knockout mice, the enhancer knockout mice show no defects in central nervous system development. They do not experience perinatal lethality. They're actually healthy and fertile. They have no impairment of B lymphocyte maturation. And B7A expression is only lost in the erythroid cells, but is maintained in the other cell types. And the hematopoietic stem cells of the enhancer knockout mice show no defect in donor reconstitution, unlike the hematopoietic stem cells of the B7A gene knockout mice. So together, these data suggest that B7A is a pleiotropic transcriptional regulator that plays numerous roles, including in neurons and B cells and hematopoietic stem cells, but that its erythroid expression depends on this intronic enhancer that itself is subject to naturally occurring disease-modifying genetic variation, and that in erythroid cells, B7A serves to repress fetal hemoglobin and thus is permissive for the disorders of adult beta globin. So this led us to a relatively simple therapeutic vision that one might collect hematopoietic stem cells from patients with hemoglobin disorders, disrupt the B7A erythroid enhancer by gene editing, and autologously reinfuse the cells. Oops. Could I have the next slide, please?
Frozen.
Well, I like Jacob Korn's postdocs too.
You will get some extra minutes at the end.
Okay. Yeah.
Okay.
Oh. Okay. Close your eyes. Great. So, well, yes, genome editing we've heard about, and I'm going to focus on the CRISPR-Cas9 system. And just to remind you, this is a system where the endonuclease Cas9 recognizes these short protospacer adjacent motif sequences shown here in yellow, and then in the presence of a complementary guide RNA with precise Watson-Crick complementarity to the adjacent genomic target, executes a double strand break. And then in this slide, you've already seen the outcomes following the double strand break depend on endogenous cellular repair mechanisms and simplified into mechanisms that lead to gene disruption on the left that rely on non-homologous end joining and generally result in small deletions of 1 to 10 nucleotides around the cleavage site, and then gene repair mechanisms that are templated and can result in precise correction of disease-associated mutations like the Sickle mutation, but that these are not necessarily co-equal pathways, and that in many cells the gene disruption mechanisms may be dominant, and that could be for a variety of reasons, as discussed in the prior talk, but at least one reason is in quiescent stem cells, like hematopoietic stem cells, that some of the templated repair mechanisms may be cell cycle dependent. So we were motivated to explore gene disruption inhibition of B7A erythroid enhancer as a potential means to therapeutically induce fetal hemoglobin. And so when we use CRISPR-Cas9 in these HUDEP2 cells, which are adult stage immortalized human erythroid precursors, in the absence of either, the BCL11A gene itself or deletion of the composite 12 kb erythroid enhancer of BCL11A, we saw dramatic loss of BCL11A expression and induction of fetal gamma globin and fetal hemoglobin protein. However, producing a 12 kb deletion was a relatively inefficient process, so we were motivated to ask the question, could single cleavages at this enhancer site be sufficient to disrupt the enhancer's function? And to do this, we conducted an agnostic screen in which we designed all the guides targeting the BCL11A erythroid enhancer. And so this is a schema of the screen drawn to linear scale for a prototypical 100 base pair enhancer element, and per 100 base pairs, there's about 13 of these NGG PAM motifs, which restrict the cleavage of SP9. So any one guide RNA will, after cleavage, will result in a variable set of small deletions, generally ranging from 1 to 10 base pairs. So each guide results in an allelic series. And then by tiling all the guides, one can achieve a series of allelic series that can approach saturation mutagenesis in situ. So we designed all these guides, synthesized them as oligos, cloned them to a lentiviral library, delivered them to cells by lentiviral transduction at low multiplicity so that each cell would just get one guide RNA. Then we were able to perform erythroid maturation of the cells, stain the cells for fetal hemoglobin, physically sort by flow cytometry cells with high and low fetal hemoglobin, isolate their genomic DNA, amplify the guides, measure by deep sequencing the representation of each guide, calculate the enrichment of that guide in the high fetal hemoglobin pool, and map its cleavage back to the genome to produce a functional enhancer map. So here's the results of that screen. For each guide, we simply calculated a fetal hemoglobin enrichment score by the number of guides in the high versus low pool. From the left, we observed a set of negative control guides showed no enrichment, and a set of positive control guides targeting exonic sequences of BCL11A showed positive enrichment. Then the finding was that most of the guides at these three enhancer sites showed no enrichment, consistent with the notion that producing mutations and most non-coding sequences would be without functional consequence. But we observed these stripes of co-localizing guides with shared effect, including this robust stripe at the plus 58 DNA hypersensitive site, where we had 10 guides that specify cleavages within 42 base pairs, whose fetal hemoglobin enrichment exceeded that of the median positive control exon targeting guide. So to summarize, this agnostic screen identified a critical functional core at the plus 58 enhancer site that had been marked by GWAS and identified sequences required for fetal hemoglobin repression. We took advantage of deep sequencing cells exposed to these guides to identify those indels specifically associated with fetal hemoglobin control, and identified that there was a core half EBOX GATA motif, which is the known binding site for the critical erythroid transcription factors, Tau1 and GATA1, whose presence was required for expression of BCL11A and repression of fetal hemoglobin. We delivered to CD34 hematopoietic stem progenitor cells Cas9 and a single guide RNA targeting the plus 58 enhancer core, and observed reduction in expression of BCL11A and induction of gamma globin and fetal hemoglobin in a manner that did not impair erythroid maturation as measured by gene expression, immunophenotype, or cell morphology. So these studies supported an idea that the erythroid enhancer might be an Achilles heel of B-sylvan-A, a mechanism whereby this key repressor of fetal hemoglobin could be therapeutically targeted. So here, the therapeutic vision would be to, by editing gene disruption, impair this core enhancer sequence, lead to reduction of B-sylvan-A expression in erythroid cells, thus leading to induction of fetal hemoglobin. This would spare the expression of B-sylvan-A in non-erythroid cells. So I want to put into context, there's actually quite a panoply of curative approaches now for the beta hemoglobin disorders, and what I just discussed is but one of them. There are approaches that rely on a variety of hematopoietic stem cell sources, from allogeneic to autologous sources. There's conventional gene therapy with addition of globin genes. There's modified gene therapy approaches that intend to induce fetal hemoglobin. And then within the genome editing space, there's a variety of approaches, some of which would intend to induce fetal hemoglobin, such as disruption of the erythroid enhancer B-sylvan-A, or creation of hereditary persistence of fetal hemoglobin alleles. And then there are gene repair strategies that could include homology-directed repair or even other repair mechanisms like base editing. So I wanted to just briefly highlight some of the other approaches, not to really do any one of those approaches full justice, but to give a sense of the landscape of strategies and some of the various advantages and challenges that these approaches may face. In fact, editing the B-sylvan-A enhancer is not something only limited to, of interest in my lab, but Sangamo and Biogen have pursued this target as well and recently published a study looking at zinc finger nucleases that target B-sylvan-A. And here they compared targeting coding sequences and enhancer sequences of B-sylvan-A and could identify zinc finger nucleases delivered to CD34 cells as mRNA that were potent in terms of generating indels, either targeting coding or enhancer sequences, and they could observe for both induction of gamma globin. But what they found here on the right is that unlike targeting the coding sequences of B-sylvan-A, which had a negative impact on the ability of the cells to persist in vivo, that the enhancer-targeted HSCs were able to persist at high levels in vivo, suggesting that targeting the erythroid enhancer may spare the hematopoietic stem cell functions of B-sylvan-A, which have been demonstrated by a number of labs now. So an alternative NHEJ strategy would be to recreate naturally occurring hereditary persistence of fetal hemoglobin alleles. These are conditions in which individuals, like the name implies, maintain a high-level expression of fetal hemoglobin throughout life. And there's two main classes of these, so-called non-deletional or deletional. The non-deletional stem from mutations at the gamma globin promoter sequences themselves. This is work done by Mitch Weiss and colleagues. And what's shown here in this box is a naturally occurring 13 base pair deletion at this gamma globin promoter. So what Dr. Weiss and colleagues did was introduce to erythroid cells Cas9 and the guide RNA, and we can focus on this one, guide RNA1 cut here. And so this is just a single cleavage, and they saw very potent induction of fetal hemoglobin after delivery of this guide RNA. What was perhaps the most remarkable aspect of this study is the most frequent allele that they retrieved from cells that got this guide RNA was actually this precise 13 nucleotide deletion, which was naturally, when naturally occurring, is associated with hereditary persistence of fetal hemoglobin. And if you squint and look closely, you can see there's microhomology surrounding this site. And so this repair event appears to be one of microhomology-mediated repair, and suggesting that, you know, simply the term NHEJ doesn't do justice to the varied pathways the cell uses to resolve double-strand breaks. But what I think is a major question that's outstanding of this work is, what is this repair efficiency of this microhomology process in hematopoietic stem cells? It may not be a process that is equally favored in HSCs as non-HSCs, and so I think that needs further investigation. Another NHEJ strategy is to mimic these so-called deletional hereditary persistence of fetal hemoglobin alleles. Here is listed a number of these deletions that occur that encompass the beta globin gene, but also surrounding sequences at the beta globin cluster, but leave gamma globin genes intact. And this is work from Y.W. Kahn and colleagues, and they focused on this HPFH5 deletion in part because it's the shortest of the deletions, about 13 kb. And so their goal was to cut out this 13 kb of sequence that included the beta globin gene but left the gamma globin gene and some regulatory elements intact, and were able to observe a dose-dependent induction of gamma globin expression upon deletion of these sequences. I think here a main question is, how efficient is this process, particularly in HSCs, and that there are anticipated competing alternative genomic outcomes when one introduces more than one cut, that there can be indels at each site, there can be inversions as well, and the functional consequences of each need to be considered. And then there's a variety of approaches to correct the sickle gene mutation, and Jacob beautifully described this work, so I'll just quickly say that this has been the work of a number of labs. Initially, Megan Hoban and Don Cohn's lab led studies looking at zinc finger nucleases to correct sickle mutation by HDR, and observed some of these alleles in the input cells at reasonable frequencies. At the same time, many indels were found in these cells, and the indels exceeded those of the repair alleles. And then particularly what happened when the cells were engrafted was that the fraction of modified repaired cells was less than 1%, whereas there was about a 23-fold advantage of non-homologous end joining disruption alleles as compared to repair alleles. And so I think this is the theme of sickle mutation repair that needs attention, is what is the repair efficiency, not just in bulk population, but in true hematopoietic stem cells. And then if there are these co-occurring high rate of indels, these are actually alleles that would be anticipated to produce beta thalassemia. And then you get an interesting possibility of a mixed hematology where a patient has cells that cause sickle cell disease, cells that may be corrected, and cells that cause thalassemia, and how those, what balance of those three it leads to either ameliorative outcome or a new disease needs attention. And then this is the work of Jacob Koren, again showing, in this case, even higher rates of HDR, but the same issue, that the engrafting alleles were substantially lower than that seen in the input cells, and that the non-homologous end joining alleles were really the dominant feature of the long-term engrafting cells. And then finally, Matt Porteous and colleagues have shown an ability to do HDR using AAV donors, and in this case, the AAV donors carry a GFP reporter, and the finding was that if one uses unselected cells, again, one can get persistence of these repair alleles, but at relatively low frequencies in vivo. But if one selects the cells that have the repair up front using either GFP sorting or other strategies, that one can then get almost all the engrafting cells to have these repair alleles. So that would be very exciting. I think what needs further attention is how would one clinically translate enriching an edited HSC population that certainly would complicate the cellular processing of what's already a somewhat complex cellular process. Okay, so some generic thoughts about how we can translate gene editing to the clinic and what are the near-term prospects for translation. Any strategy to get to the clinic needs to be robust, needs to have high editing efficiency at a clinical scale. What we're talking about here, hundreds of millions of CD34 cells need to be edited, and so we need reagents that are clinical grade. But I think the feasibility of this approach is underscored by ongoing work in the field, including that from Sangamo and City of Hope, looking at editing the CCR5 gene in CD34 cells for autologous transplant of HIV-positive patients. And we may hear more about that trial later at this meeting. Specificity, of course, is important. The major toxicity that one considers with genome editing that's intrinsic to the genome editing is any genotoxicity. There's a bit of an arms race going on with increasingly sensitive detection methods and increasingly specific reagents. But at the end of the day, one may be left with off-target mutations of unknown and perhaps no biological significance. And it may even not be possible to detect every rare allele. One can imagine at the extreme end, there may be an allele that either one measures or one puts into a patient, but can't do both. And that I think I would fully agree with Jacob's comments that functional assays are necessary, but the challenge is that the functional assays of modified HSPC products are really of uncertain clinical predictive value. And so we really need better ways to test these cells and predict what they will do in patients. One needs to isolate the cells. That's kind of obvious, but a unique challenge, especially in sickle cell disease, is that the conventional form of mobilization by GCSF is contraindicated, can precipitate sickle crisis. But there's hope. There's ongoing trials of plerixafor at numerous centers as a means to obtain HSPCs from sickle cell patients that doesn't require bone marrow harvest, which typically has been a sedated procedure requiring multiple anesthesias to get adequate cell numbers. The gene-edited cells have to engraft. This typically relies on myeloablative therapies, but there's great opportunities for the development of less toxic conditioning regimens. And in the past couple of years, some very exciting avenues have come forward with antibody-based therapies. That really, although one edits CD34 cells, it's the rare hematopoietic stem cells in that pool that need to be edited, and the current assays are imperfect. The mouse xenograft models are the best we have, but they're really not perfect measures of human HSC function. That there may be roles for non-human primates to allow the assessment both of erythropoiesis in vivo, which is not possible in the xenograft models, and also the HSC function following an auto transplant. However, one can really never measure human-specific off-targets in a non-human primate model. And then there's really great opportunities here if one could better isolate, expand, and generate hematopoietic stem cells. That we don't need 100% fetal hemoglobin, we don't need 100% of cells edited. We know from naturally occurring mutations that about 30% fetal hemoglobin in a pancellular distribution prevents clinical manifestations of the hemoglobin disorders. We know that haploinsufficiency for B7A results in hereditary persistence of fetal hemoglobin, so even a twofold reduction in B7A levels would be very potent. And we know from the results of clinical observations of allogeneic transplant with mixed chimerism that just 10 to 20% of non-sickle or non-thalassemia hematopoiesis may provide clinical benefit due to the survival advantage of the healthy donor erythroid cells. And this is one example where 11% chimerism gave greater than 90% non-sickle hemoglobin. And then finally, I think the future opportunities would be if we had better ways to produce, expand HSCs, to obtain engraftable products from pluripotent stem cells, or if we could achieve in vivo delivery that would greatly simplify the complicated ex vivo cellular processing and might even allow redosing. So the summit is cure of the beta hemoglobin disorders. There's really multiple plausible paths to achieve this summit. We need to explore all of these to determine what is most safe, effective, and what can scale to as many patients as possible in the United States and around the world. So with that, thanks for your attention. Sorry for the technical difficulties. I'd like to thank some colleagues, collaborators, and funders. But in the interest of time, I'll stop here and take any questions.
Thank you very much, Daniel, for this great overview and for sharing your thoughts with us. We have time for one or two questions from the audience. Maybe I start with the first one. I think you nicely showed that NHEJ can lead to, let's say, gene modification efficiencies of maybe 70, 80, or even 90%. But I think it's harder to predict the outcome of a genome editing event. So have you looked into the variability and also reproducibility of your genome editing at the locus?
Yeah, so I think so one key issue in genome editing is when you have the non-homologous end joining, you get a spectrum of indels, but emerging evidence is it's really non-random and that there are some predictors, although imprecise, of what sort of alleles are produced. I think for the NHEJ, we haven't seen lots of donor-to-donor variability. Most of the variability that we've observed is guide to guide, but that within a guide, we typically get a quite stereotypic pattern of repair. I think one issue is how many of the repair alleles would be predicted to be protective. And the goal for NHEJ would be to have as many of the alleles produced as possible be ameliorating. Yeah.
Okay, thank you very much, Daniel. We move on.
Yeah. So next I'd like to introduce Dr. Laurie Solo from Northwestern University, and she's also the next dean of the Divinity School of the University of Chicago. She has a long and distinguished career as a bioethicist, and she also served on the RAC, the Recombinant DNA Advisory Board. And today she will talk about what actually happens once we get genome editing to work and what the ethical implications are in the future.
Okay, thank you. And I need my slides. I could just talk, but I know you like slides, right? Okay. So I'm going to talk about the ethical issues in germline gene editing, which we haven't addressed yet today, but is the obvious implications of what we're thinking about. And see, I'm a humanist, so I'm really having trouble with this. This here? Okay, better. Okay, this is my conflict of interest statement. I have none, right? I'm sort of like looking for conflicts, but all right. I want to start with a history of how we regulate and how we think about human gene editing. And this is a picture of Maxine Singer. How many here actually knew Maxine Singer? She's the hero for bioethicists because she was able to bring the first questions about the ethical implications of this powerful new technology using viral vectors to edit genes. And she sent this letter, Dieter Sohl and Maxine Singer worked together, and looking at this letter, this was sent to the national academies to say, wait a minute, we have questions that are enormous about this new power that we're discussing in an early Gordon conference. And she put together this call. And look, you can see the scientific developments over the past 2 years, look at the short timeline, make it both reasonable and convenient to generate overlapping sequences bacteria and animal viruses, such sequences, right, on and on, so that they, using Watson-Crick bonding, this is just the first foray into what will become your association. This is 20 years before the timeline starts. In your description, your self-description. And here's a very early warning call saying we have a new source of power, but what's the implications of this power? Have we gone too far? And this call actually was met with a lot of alarm. In Cambridge, Massachusetts, there was a city council meeting, and the city council meeting depicted here meant that no DNA could be edited in Cambridge, Massachusetts. That stayed for a very long time, actually. And people met at Asilomar. Now, Asilomar, for those Californians, is a windswept, somewhat primitive, state-funded meeting place put together by Julia Morgan for young women who wanted to think about higher education. And people met in Asilomar, and everyone who thought about gene editing could still fit in one room. It was all the scientists, you can see Singer right there, all the scientists, a few press people, a couple of bioethicists, and some lawyers who thought we have to begin to think about the regulatory power that would be the limiting steps for this new scientific power. They spent about a week there, and they understood that unless they came up with self-regulatory solutions, Congress would begin to regulate it without them. And they were worried about these things. They created basic safety rules. And at Asilomar, there was a move away from larger ethical concerns to smaller concerns about safety. And Asilomar put forward these 3 rules. Don't insert pathogens into your E. coli. Don't insert cancer sequences into E. coli. They're afraid of spreading. And don't insert genes for drug resistance. See how limited the safety concerns were 1972-73. All the work had to be done in a level 4 biocontamination facility, which you find amusing now. Actually, one of the facilities at Johns Hopkins is now used as a storage room. It still has the old writing across it, so you can go take a tour. It was banned in Cambridge, as I said, and the idea was that science self-regulation was put together just in time because Edward Kennedy was about to go forward in Congress with a bill regulating human gene editing of E. coli, and just before he put that bill forward, the capacity to make artificial insulin, human growth hormone was developed, and people raced in and said, we will regulate ourselves. Congress, step back, we'll have self-regulation. And of course, there was much literature about it then. Leroy Walters, who was at Asilomar, one of the first bioethicists to take a look at this, said there's critical concerns about human gene therapy, but by and large, it should go forward under certain conditions of restraint. And he wrote what was called the points to consider, and if you've gone to the RAC, you've seen the points to consider as part of your application. And the points to consider there's a gentle way of regulation, just saying think about these things before you go forward. One was that ethical issues had to be a part of research on recombinant DNA, and this began a long series of a small inclusion in big science projects, 3% in most science projects, including nanotechnology and the human genome itself. The idea would be that safety would be the regulatory lever for addressing concerns. The RAC was formed, the first national body to regulate all research, and that led to the Appendix M, the points to consider, which you've all filled out. And the RAC also led to biosafety committees and guidelines for accidents in the lab. So beginning the regulatory structure early on in 1972. And these were the concerns that Walters put forward, and they were the same questions that we ask of every researcher that comes before the RAC. What potential harms and benefits to research subjects would there be? How will potential harms and benefits be communicated? So much emphasis on the informed consent form. And how will selection among research subjects be made? And how will privacy and confidentiality be preserved? You can see what's not there is anything about commercialization or patenting or benefits or who would pay for side effects, but notice the enormous emphasis on the consent form. If you've come before the RAC, especially if I've been on the RAC, you'll know that the bioethicists, my colleague here knows this, the bioethicists focus on what does the patient know? How is this human subject complicities being regulated? Does the patient fully know what a phase 1 clinical trial is? But that was not the only thing the public worried about. While we in the RAC and the scientific community, the bioethics community, had felt that regulating for science and for autonomy and consent was the key, the public consistently raised other objections. And here are 10 canonical objections to anything with the word genetic in it or gene in it. That was a broad objection to genetic alteration. And here are the reasons that people put forward. And I want to say that these were all put forward in 1972. They were raised in Leroy Walter's book, and some of them are still enduring for this year's debate. First was that something about the DNA itself shouldn't be altered. It was the really real being of any ipsity, of any entity. People talked about DNA in the popular press. Even President Obama talks about it's in our DNA, for instance, in the way that medieval theologians talked about the soul, that it was the real essence of the being. And so given the fact that they talked about it as the soul in this medieval way, it began to acquire this sort of semi-spiritual quality. And you'll see it everywhere, this notion of now my real identity is revealed, and all those ads for DNA test kits where the person changes their outfits and suddenly becomes Scottish or something. It's because this DNA is their real soul, their interior ipsity. And notice that goes forward. My university, Northwestern University, suggests that in fact the word "and" is in the DNA of a Northwestern student, for instance. The second objection is that nature is normative, meaning it gives you rules for behavior. This is natural law theory. Nature is normative, and that nature is good and fixed, and human beings should not alter nature. The third is that suffering and mortality defines our humanity. And there was a concern that was raised about the drive for perfection that DNA repair might be. The fourth is that slopes are slippery, and once we start to do one small thing, beta thalassemia, for instance, it would lead the way towards enhancements, and enhancements might create significant divisions between human beings. Fifth is that dual use was inevitable. There was a concern in 1970. We were still fighting the Cold War. Now we worry about Chinese people making strange warriors out of DNA, an actual question that was put before the National Academy, that dual use, weaponry use, would be inevitable. 6 was that error was inevitable. 7 is about marketplace. Little did they know what would happen to the marketplace in the ensuing 20 years. But even in 73, people began to worry that the marketplace would drive and corrupt science. The 8th objection was that the world was already unfair, and that this sort of manipulation of human genomes would create 2 classes of beings, wild types and people who were designed, and that this would create and worsen injustice in our communities. 9 is that the world was already inauthentic and unheimlich, different and altered enough. And 10th is that finitude and contingency were our defining human values, not mastery, not control. And there was much talk in the literature about mastery as an error of human judgment. Now, those objections, if you look at them carefully, and it's not just me who's a theorist of religion, those are semi-religious objections. They deal with what might happen in the future. They're eschatological in their thrust. And in 1999, scholars of religion, Ted Friedman, one of your heroes of your organization, scientists and lawyers got together at the AAAS and said, what would be the real objections? Not the fantastic objections, not the science fiction objections, not the Martian objections, but what are the real problems with editing, making inheritable genetic mutations? This was the first time that the different groups with different ideas had sat down to have a policy-driven discussion. And I was on that discussion, and here's what we thought in 1999 when we were convinced, because you told bioethicists this, that you were just on the cusp of using viral vectors to drive germline mutations. The first one was about principles. What's the principle that should define your work? And are there reasons in principle why performing basic research should be impermissible? Is this in the category of the kind of knowledge that we just shouldn't know? Is there any knowledge in that category? And is this germline heritable mutation that sort of knowledge, an impermissible knowledge? The second question is about the contextual factors had to be taken into account, and worrying about the contextual factors that might prevent development. So, for instance, we live in a society that is riven by racism. It's the subject of debate on and on and on. Would physical difference, with creating physical difference, is that such a difficult discussion for Americans that the context of this work would be impermissible? We just couldn't, in this particular society, do this sort of work. Maybe in another society, but not in our society. Third would be the type of work you would do. What are you going to alter the genome for? right? Would it be for trivial things? Would it be for serious things? What's the sort of purpose that you would use? And finally, how are you going to make it, how are you going to regulate it? How are you going to regulate the structures? How are you going to set up the procedures? Who's going to be in charge? Should it be done by the state? Should it be done by the profession itself? Should it be done by people like philosophers and theologians? And these questions became the guiding questions for the continuing debate on your work. Now, because of this, there was a consensus, and this is one of the strongest international consensus, both in my field and in the scientific field, and in the policy field, that somatic alterations are permissible, but inherited germline alterations were not permissible. And secondly, that prevention and treatment of disease was permissible, but enhancement was not. And these two consensus, these two bright lines, now if you're a scientist, you know that there's some slippage in these things, and it's very hard to make the bright lines in science, but they were surely made in international policy. Okay, so what is new? Why this year are we reconsidering this? Here are some reasons. First of all, the viral vector idea was complexly flawed. That's why we kept having imperfect results. It was flawed with terrible off-target effects and disruption of the immune system. The classic story and tragic story of Jesse Gelsinger's death knocked the field off in another direction. People began to worry about certainty. And thus, fingers and talons and CRISPR-Cas9 began to be developed. And there was a turn towards new tools of synthetic biology by engineers, by the way, whose experience is sort of orthogonal to clinical medicine, but whose tools look powerful and interesting. And, of course, there was this. You notice that the clothes got better 20 years later, right? There are 7 new things that drive the debate now. First is the marketplace is very different in 2017 than it was in 1973. Both in small ways and in large ways, with immediate conflicts. The idea of a product, the idea of a patent, the idea of an ownership. What's a decent sort of profit to make? Can conflicts be managed? Is a little slide enough, right? Jonas Salk invents polio vaccine and doesn't patent it, because patenting, when Salk developed the vaccine, was unthinkable. He said, you might as well patent the sun. After 40 years, we've had significant failure. We're a less optimistic field. There's a hope and a hype with the same group of people. Everybody that was, many, many people that were there in that room in the Asilomar still are part of the field. And we've heard, bioethicists who's watching, lots of promises. I call this the decophilia promise, the problem with decophilia, that's love of 10 years. Like in 10 years, we're going to have it in clinical trials, which I have heard since 1973 when I started, and I no longer believe. There's been a growth of patient advocacy, activism, starting with AIDS, internet connectivity, so that the public knows much more and thinks it knows much more about how to run science. There is a deep and fundamental unknowability that now is really unfolding. Look at the 2 talks you just heard. Look at all the blank spaces. This acknowledgement of the deep, the fundamental unknowability, the not yet there-ness, is a part of science that wasn't there in the more optimistic 1973 period. The world of environmental catastrophe has happened. There's been significant mistakes. People have concerns about science because there has been huge science failures. There's, of course, been the frauds of stem cell research. That has shaken public confidence as well. There is a reality of terrorism that didn't exist in 73, and yes, scientists with DARPA funding do get engaged in that discourse. I was part of a NIS panel that looked at the Department of Defense query about whether advanced reproductive technology could be used by the Chinese in an offensive way, and whether we should use it in a defensive way. And then finally, there's a new enthusiasm for high-tech solutions for fundamental realities, such as aging. And because funding has shifted from the public to the private, one of the biggest new things in the marketplace, because then the funding is driven by individual biotech millionaires who, for instance, don't want to get old and fund whole projects about not aging. None of these things were a factor in the Asilomar debates, not one of them. So the National Academies of the US, UK, and China in 2005, December, had what they called, very self-consciously, an Asilomar-type meeting. As I said, the clothes were much better, much more fancy high-tech. I was at this meeting, and the conclusions of the meeting was these 4 things. 1, that basic and preclinical research was permitted, and as you know, rapidly began in the UK. 2 was that clinical use was permitted for somatic ending. The somatic cell idea passed the RAC review in June, just barely 6 months later. 3 was that in clinical use, germlines was not permitted at that time because it would be irresponsible to proceed with any clinical use of the germline technologies. And then the 4th was calling for an ongoing forum and a committee to discuss these 4 points. Now notice the power of the thought that it would be irresponsible to proceed with any clinical use of germline editing. This was emphatically stated and largely to try to avoid non-state actors from using this technology, but also to create a framework for what would be these criteria, how to create this broad social consensus about the appropriateness of the application. And then, in February of this year, Valentine's Day, the National Academy's report was released, reopening the debate as the first of the opening 4 that was called for. Is it still justified, they asked, to prohibit the use of editing for heritable conditions? Can we now alter the genetic fate of our descendants? And they said, let's look at 2 sorts of competing principles. Notice the reoccurrence of some of the canonical objections from 20 years ago. First was that we live in a world of fundamental injustice, a fundamentally unjust world in which the wealthy already can access social goods for their children and make their children more designed than people who are ordinary, people who are living, for instance, in sub-Saharan Africa, and that fundamental injustice is unfair, and we should not potentiate it. Secondly, that human beings are begotten and they're not made, that something about our humanity, our humanness, is part of the equal participation in the great gift of being human, a fundamentally religious objection. An interesting objection by Gilbert and Melander said that we're all equal in that we are not one of us is the project or property of another. And because we're not obliged to each other in this particular marketplace way, this allowing people to design their children would shift this fundamental relationship of equality. And finally, and very powerfully, we live in what's called in bioethics the shadow of history. The shadow of history refers to the crisis of the eugenic movement that led up to, first, the categorization of Jews as mutationally different, and then as dangerous to the state, and then as subjects of elimination. The shadow of history shows us that when the state uses genetics to create what they think is a better future, a healthier future, that it can be used to destroy whole populations. That shadow of history still shapes all of your work, and quite frankly, always should. That question should always be a part of this sort of project. And given the fact that things were not going well this last year in terms of human love and mutual respect, this shadow of history had tremendous power. The other side made these objections. We should permit this research. Why? Because the telos of medicine, the ultimate fitness, the ultimate goal, this is an Aristotelian term, means that medical research, medicine and medical research, their whole function is to address human suffering. That's the whole point of whatever you do. You don't do it for any other reason. To enact this telos fully would mean going to the next step and to eliminate the disease-causing alleles forever. Like, why should you maintain them? And finally, that we have done this before as a species. We have allowed vaccination. We have allowed statins. We have allowed antibiotics. We've wildly changed how long people live and their relationship to one another through science. And we've already made a sort of selection. And this is just the continuation of that moral gesture. And by the way, these are both utilitarian concerns and deontological concerns. It's not just that it's the greatest good for the greatest number, or you really want the consequence to be good. It is also that as scientists, you have a role-specific duty, a deontological, duty-based, role-specific duty to avoid human suffering and to treat and to cure. And that your work itself should be driven by that duty. And that's ultimately what won. That was an argument that the National Academy's IOM found persuasive, that they had a new consensus report that came out to proceed with this work with caution. It's called the yellow light response. These are the overarching principles of that report, that germline genetic intervention should be allowed if it promotes well-being of the individual and of society, if it's done under conditions of transparency, if research subjects are treated with what's called due care, if the scientific rules for responsible conduct in research are upheld, if respect for persons, for the human dignity of individual persons is organized, well organized, if it creates a world of more fairness and not less fairness, and if it is founded in transnational cooperation. These are the principles of the national report. And what's interesting about the report is scientists and bioethicists said there is a lot of existing oversight, and this is the case. The RAC is still there, FDA is still there, laboratory research for cells and tissues has robust oversight, review and evaluation of somatic gene therapy by all the existing regulatory agencies is still quite strong, and the oversight for the new off-target effects, the genetic location, other factors, that's already in place. So no new thing needs to happen for that, but new oversight is needed for some specific uses of gene editing. One is they gave no approval for clinical trials of gene therapy that's not intended to treat or prevent disease or disability. So no enhancement, but only for severe genetic diseases and severe disabilities. The work had to be transparent, and inclusive public debate should precede any consideration of whether to authorize for any other indication, meaning anything besides severe disease or severe disability. So those are the two kinds of oversight that needed. So these are no's on those two things. And this is the critical recommendation of the many they made. Recommendation 5.1, the clinical trials would be permitted within a robust and effective regulatory structure. And this 5.1 recommendation sets the rules for your work. First is there has to be the absence of any reasonable alternative. We were just given two examples of the trouble treating a very widespread genetic disease, the ineffectiveness of existing treatments. There has to be no other reasonable alternative for patients. Two is the restriction, it has to be preventing a serious disease or disability, and the disease has to be quite significant. They didn't restrict it to only fatal diseases like Tay-Sachs, but they thought significant and severe disease and disability. Three is it was restricted to editing genes that have been convincingly demonstrated to cause or strongly predispose to the specific disease. So you have to really know what your target. Fourth was it had to be restricted to converting these genes to versions that are already in the population. In other words, the harmless alleles, the non-disease producing alleles, and they're known, the ones that were known to be associated with the disease but have little or no adverse effects in the non-disease population. The availability of credible preclinical and clinical trials on risks and benefits should precede the procedures. They should be demonstrated. During the trial, there should be ongoing, rigorous DSMB oversight on the health and safety of research subjects. There should be comprehensive plans for long-term, multi-generational follow-up that still respects personal autonomy. Notice they've split the difference there between protecting long-term subjects, but notice that there's no funding for this, but there's a call for this. There has to be maximum transparency, public transparency coincident with patient privacy. There has to be a continual reassessment of both health and societal benefits. Notice not just health benefits, this is a difference. Health and societal benefits and risks with broad and ongoing participation and input from the public. There has to be a reliable oversight mechanism, not defined, to be developed later to prevent the extension of other uses other than preventing a serious disease or condition. So there's going to be some unnamed, to be named later, mechanism to keep this technology in the hands of serious scientists who are willing to abide by these rules. And there was finally a recognition of the difficulty in the American current political landscape with this administration, both the president and certainly the vice president, that actually would restrict any use of federal funds for anything using embryo, any germline or any embryo research at first. So they were aware of the complexities, but they were writing for the long arc of your scientific research, not only for this particular moment. They wanted to set in place things for the future. They call for robust community discourse. They use the example of European nations such as Denmark and France, which have had these community discourses about such things as reproductive technology or the use of nuclear energy, as in France. They called for an ongoing reassessment of if this community discourse is working, to make it inclusive, continual, and to have regulatory power. Notice the shift from the regulatory power within the scientific community to the regulatory power emerging out of this deliberative discourse, this public reason. And I want to say that this is one meeting in which I think we have begun. You are part of the community, and you will be, in fact, the leaders of such discourses within your own communities. I think these 4 questions are still in play, and I'm someone who says to my students when we debate emerging technologies, I say in my classroom, there's no Martians, no Nazis, and no robots, that you have to focus on the real questions that scientists grapple with every day. And I really want to raise these 4 questions to you because I think these are the ones to think about when you think about the ethical standards of your profession and the regulation of your very exciting and very optimistic research. Can you think of a reason why there's a principle against doing inheritable germline modification? Is there a principle that you can come up with? And if so, what is that principle? And of course, we suggested too, the notion of mastery and the notion of equality might be principles in play. 2 is that what contextual factors and what's your role in thinking about these contextual factors? Is the United States of America the proper place to have this kind of research, or is it too fraught here? Is there any ideal community in which this could be used as a test case that wouldn't have the conditions that we face here? When I think about the sickle cell, the elegant sickle cell work that you're doing in your labs, and I think about the realities of sub-Saharan Africa and how you can't get a glass of clean water in a third of the world, I worry about that as the context for your elegant research. And how do we do that, especially in a country that just can't decide if we're going to have health care for everybody, much less this sort of complex health care? What purposes and what techniques and what applications will be permitted? Yes, we can agree on using this to inhibit fatal illnesses, but what about more subtle modifications? Would it ever be permissible ethically? And why or why not to make the human species kinder, smarter, more complicated, if that could ever be figured out? And what would be the reason against those ideas? What is that really? Would you stop research intended for good, and why? And finally, who decides? Who do you want to be in charge of this? The stem cell community, a community that I've been a part of for many years, has struggled to try to keep fake stem cell clinics out of business. That's been an intellectual project for a decade. And yet you can open any paper in this country or in any other country and see advertisements for stem cell clinics as if they're real. How can this very powerful technology ever be regulated with the same kind of rigor that you'd want to prohibit the harmful societal effects? Now, your questions, because it's the beginning of a robust dialogue, and thanks. Thanks to all the scientists who help me understand this, who teach me what the acronyms mean. And thank you very much for the invitation to talk with you about this new National Academy's proposals that we hope is a good and thoughtful way to think about gene editing in the future. Thank you.
Questions, please? So maybe get started. How do you see these regulations like in the international context? Obviously, the initial NAS meeting was very inclusive, Europe, Asia, and there's still countries out there, especially in Europe, where basically germline editing still is like banned at this point.
The last principle, as you note on that list, was international cooperation. And yet, ironically, this was a step forward by the United States regulatory community against an international consensus. There is, to date, a very strong consensus against doing heritable germline editing. And this is the first challenge to that discourse. So, but theoretically, they were calling for this to be the new rules for everywhere. But yeah, that's yet to be unfolded.
I guess that was Cindy Dunbar, NHLPI. A little confusing between, what happened between December 2015 when it was irresponsible to think about germline editing to, February. I mean, many of us were sort of, who weren't actually involved in what went on, and there's all kinds of theories and conspiracy theories of all sorts of kinds, not conspiracy theories, but you know what I mean. And it's very hard for those working in somatic editing to kind of deal with the whiplash that this created, especially in the U.S. in the current political context. So I wonder if you have any insights of how we sort of answer people who are coming to us saying, well, I thought you said you weren't going to do germline editing, and now you are.
Yeah.
What's going on?
You know, I love that you asked that question because I'm a bioethicist, and I had the same feeling, like, you promised you couldn't do mammalian cloning. Like, what do you mean you can do it, right? You promised, and I feel, and this was the same thing. We spent decades achieving a strong international consensus about those bright lines. And I thought we had it nailed down in 1999. I thought, like, okay, wide-ranging research on somatic interventions, get it right, learn how to do it, and then and only then move forward. Essentially, I think, so I struggled when I read the report, like, what happened, right? In part because the people on the committee were people who were excited about this potential, who couldn't find intellectually good reasons, either scientifically or legally, to make the bright lines hold, given the scientific problems with them. I don't need to rehearse here. And also, they had an odd paragraph about transhumanism. Now, I'm a scholar of religion, right? And so I was struck by the fact that when I Googled word search religions, there wasn't much on what religious communities would say about this. There were two speakers, there was Gilbert Melander and Michael Sandel, who did raise the question about mastery that I mentioned here, but I thought were powerfully interesting problems, and they come from a religious sensibility. But the word transhumanism reappeared several times as if those voices should be part of the discourse. And they were very much a part and speakers at the National Academies meetings, as you know. And so that's a new idea. Personally, I think it's a kind of silly idea. I like being human without any modification there. I think we're a good species. We need to be humble about that. But the transhumanists have made this debate that we should be better, we should be pushing forward. That's logical positivism in its strongest form, and you can see its optimistic glitter throughout that report. So I think that pushes it. I do think the reality is that the two bright lines are going to be guarded pretty intensely in the United States by the budgetary constraints on embryo research. And I think in other communities as well, though there have been indications that the British might be going ahead in this direction. So there's a lot of fences around this notion, right? There's a lot of constraints to keep this very narrowly focused and to move ahead very, very cautiously. Yes?
Hi, Juan Pablo Ruiz. My question is, the conversations that are happening are obviously happening with people who are heading the labs or the bioethicists, and that's very important. But for those of us who are PhD students and postdocs, the younger generations who will be, having a little bit more time with the consequences of all of this, is there a spot on the table for us, for our voices to be heard? And if there is, how do we as younger students and postdocs get involved in these conversations and in this topic?
There's many calls. It's such a good question. There's many calls for this robust community discourse. And up until now, I frankly haven't seen that played out, even though at the end of every paper and every policy in bioethics for the last 20 years has called for the robust discourse. But that would mean really rethinking what the life work of scientists would be. Like, you can see how the climate scientists are struggling with this problem. Like, how do we get our science, our facts, out into the community for rational discourse at a time when facts themselves are a suspect. But I think if there's going to be community discourse, as they call for, that will set the regulatory standards, that has to be at PTA meetings and trade union meetings, right? And bowling alleys and places where ordinary people sit around and talk. And that sort of science education has yet to be developed. It's called for in the report. I think it's a great idea, but it's yet to be developed, and certainly it's not yet funded. And that you would need that money to go out and talk to your neighborhood, right? But theoretically, it should be a part of every new science project.
Okay. I've been involved in many ethical discussions in Germany, a place where genome editing in the germline is banned.
Yes.
I think the question that always comes up is, do we need germline editing if we have other alternative methods, like, for instance, pre-implantation diagnostics?
I'm with you. I think that the strength of pre-implantation diagnostics is very, very powerful. And for instance, in the mtDNA debates, when Britain went ahead and then the US and national academies went ahead as well, I was opposed to it. I thought wait a minute, we have pre-implantation diagnostics. There's no need to really repair the DNA if we can choose different options. Notice why this works, though. I don't believe that the DNA is the really real. I believe in a whole being as the really real, right? Because I'm a Jewish theologian, and we don't believe in a difference between body and soul. But there's a strong idea that the DNA is the soul. And so there's this notion that people want to have a matching DNA, nuclear DNA, to themselves, and that DNA, that is that's a being that exists, and that being needs to be fixed and then brought to life. Do you see? But that's a theological claim. And that's really an interesting part of this debate. Mm-hmm. What they want to do is to retain the intactness of a specific ipsity and fix that ipsity. That's the theory. And that theory goes against many scientists' ideas about when human beings begin to develop, right? So, yes, it's very theologically problematic to place all your ideas about identity and self into the nuclear DNA of the embryo. That is a very problematic idea theologically. And the Germans, rightly so, continue and ought to continue raising the question of the validity of that sort of project in general. Yes?
Yes. For germline editing, what's the discussion and thoughts around the issues of consent in future generations?
Right. So that's a very interesting question, especially in a world where the moral status of the human embryo is at stake. Like, the embryos can't consent, so we defer to parental consent, right? And why that's not such a bad idea is that I have 5 children, right? And I've made many decisions totally without their consent. I've decided to raise them as Jews. I've decided to live in a certain area. I've decided to vaccinate them. I've made many decisions on education, on social framing, on much of their first 18 years have been their unconsented life, right? And it's been my consent. So I do think locating it in parental responsibility, in parental love, is a far better way to locate it rather than in the hands of scientists or the hands of the government. I do think that we have to respect that bond and hope that will be... that with flaws, with mistakes, and with sets of bad ideas, but in general, parents love their children and want the best for them. And I cling to that as the best regulatory mechanism.
Okay.
Okay.
Thank you very much, Laurie. I think this concludes our session. Let's thank the